390 and it is going up to the 760 nanometer. Within this we have violet, we have indigo, we have blue, we have green, and we have yellow, then we have orange, then we have orange red, and then we have red, and after that we have far red. Okay. So, all these different wavelength lights actually are exposing the pigments which are present within the chloroplast, and that they are using it for the making the chemical energy. Okay. But by doing systematic experiments, scientists have found out that the blue light is actually containing the is very photoactive in doing the photosynthesis, because it contains more energy, whereas the red light is also doing the photosynthesis, but it contains less energy. But as far as the efficiency is concerned, the blue light, the red light, although the energy is less, the maximum amount of energy which is present with the red light is being absorbed. So, it is the most efficient in terms of the photosynthesis, and whereas in the case of blue light it is the most active, but the as far as the efficiency is concerned, it is least efficient, because the lot of energy which is associated with the blue light is being go into the waste. And this region of blue to red is known as the photoactive region, because this is the region which is participating into the photosynthetic activity, or PAR, and why the blue light has the more energy and less red energy, red light has less energy, because the energy is expressed by the expression known as E equal to h nu, which is actually the discovery by the Albert Einstein. And if I put the value of wavelength, it can be expressed like this. So, the shorter the wavelength, so if you have less, you are going to have the more energy. So, the wavelength of the blue light is lower, and it is actually in the range of 470 to 500, whereas in the case of red it is in the range of 600 to 700. So, the energy which is associated with the red light is less compared to the blue light, but as far as the efficiency is concerned, it is more efficient for the red light. Now, the question comes, although the visible light contains so many different types of visible light contains the light of different colors. So, how the photosynthetic machinery is utilizing this energy, utilizing this light of different wavelength to catalyze the reactions to convert the photo energy into the chemical energy. And that question comes when we see the absorption spectra of different pigments, what we have discussed in the previous class. So, what is mean by the absorption spectra? So, what is mean by absorption spectra is that if you plot the absorbance of a molecule against the wavelength in a 2D graph. So, what you have to do for getting an absorption spectra of a typical fluorophore or typical molecule or the pigment is you take the pigment and put it into a cuvette. Okay. So, you make the pigment, you make the suitable solution, put it into a cuvette, and then you take that cuvette and put it into the spectrophotometer, and then ask the spectrophotometer to excite this molecule with the different wavelength light and tell me what is the absorbance. So, what will happen during this particular kind of experiment, when you are exciting the molecules, the molecule is taking up this energy, and what is there in the molecule to take up the energy? The electrons. So, once the electron take up the energy which is present in the particular wavelength, it goes from one energy state to another energy state, and that actually will reflect in terms of their absorbance. So, if you take the particular spectra, what you will see is a pattern like this. Okay? And this particular pattern is going to be a signature sequence or signature pattern for the particular molecule, signature spectra for a pigment. As long as the pigment is in the state of the pigment is not changing, and as well as the solvent is not changing, means if you are making a solution of chlorophyll into the methanol, the particular pigment will remain, the particular signature spectra will remain intact until you are dissolving the chlorophyll into the methanol. But if you dissolve the chlorophyll into ethanol or acetone or any other organic solvent, probably the pattern will change, because that actually has the shielding as well as other kind of effects. So, this kind of spectra gives two information. One, that at this particular wavelength, the molecule is absorbing maximum. This means in this particular wavelength, which is actually the lambda max, the molecule is going to be most efficient, means it is going to take up this particular kind of energy into utilizing that energy for its work. The second information what we can also get if we also take a fluorescence spectra is that whether this molecule is also transferring that energy to other molecule as well. So, let us take let us see the absorption spectra of different pigments. So, what you can see is the spectra of different pigments, and what we are going to discuss is about the spectra of chlorophyll as well as the accessory molecules. So, what you can see is the spectra of chlorophyll A. So, you can see that the chlorophyll A is actually absorbing maximally in the red region, whereas the chlorophyll B is maximally absorbing in the range of in the blue, and the other molecule, for example, the beta carotene, what you can see this orange color spectra, the beta carotene is absorbing maximally in the blue region, whereas the phycoerythrine is absorbing in the range of 500 to 600 with the peak at around 550, and that actually gives the clues about how the chlorophyll machinery, which contains all these different pigments, are actually utilizing them to drive the photosynthesis process with such a high efficiency. So, what happens when any kind of light, if any kind of light falls, for example, if you keep the plant in a blue color light or in a red color light, it doesn't matter, because what matters is that as long as these pigments are present within the chlorophyll, for example, if you put it into a blue light, what will happen is beta carotene is going to absorb that light maximally and then transfer that energy to the chlorophyll and provide the energy to the chlorophyll. Although the chlorophyll is absorbing maximally in the red region, but since you are not even if you are not providing the red wavelength, it will take up the energy from the beta carotene which is present within the vicinity in the chloroplast and run the photosynthesis process. Similarly, you can understand how the phycoerythrin, which is also accessory molecule, is also going to participate in the process of photosynthesis. So, all these molecules are actually being used to drive the photosynthetic process, and that is why they constitute a complex, and that complex is known as the light harvesting complex. So, let us discuss what are the components are present in the light harvesting complex. So, as we said, the light harvesting complex are going to have a series of pigments okay. So, imagine that the sunlight is coming from this direction. So, sunlight is giving the energy. So, what you need? You need a coat of the accessory pigments, so that the molecule which are present within the core of this light harvesting complex should not get this high energy light. So, what you need is a layer of accessory pigment, and we discussed that accessory pigment means the pigment which are belonging to the carotenoids or the phycobilin, and then these are actually constituting the antenna. So, antenna's job is to receive the light, primary light from the sun, and then they will take up this energy and then transfer this energy to the core molecules. Okay. So, these are the core pigment. So, these core pigments, then they will receive this energy, okay, and ultimately these core pigments are going to transfer this into the photo center or the reaction center. If you remember our discussion about the chlorophyll, we have discussed that we have two reaction center. One is called as the photosystem 1 and other one is called as photosystem 2. So, accordingly we have two different types of light harvesting complex, and these are called as light harvesting complex 1, LHC1 and LHC2, and LHC1 actually contains the photosystem 1 and LHC2 contains the photosystem 2. So, what you can see in the PowerPoint slide that so, as we discussed, we have two different kinds of light harvesting complex, light harvesting complex 1, light harvesting complex 2. So, in the light harvesting complex 1 and the basic composition or the basic machine configuration of the light harvesting complex 1 or the light harvesting complex 2 remains constant. So, means that they have the antenna pigments on the periphery, then they contains the core accessory pigments which actually will be on the center, and then ultimately they are going to have the reaction center which actually will absorb or utilize this light energy to with the help of the electron donor or the electron acceptors. So, in the light harvesting complex 1, it exists in two different forms. One is called LSC1P680 and the LSC1P730, and these two are actually collectively known as the photosystem 1. So, photosystem 1, the reaction center, this reaction center in the case of light harvesting gun or the photosystem 1 is made up of a dimer of chlorophyll A P700, and it has a antenna complex, this the top antenna complex where approximately the 70 to 80 chlorophyll molecules are present, and it also contains 10 to 12 molecules of beta carotene. So, beta carotene is actually competent enough to absorb very high energy light, and whereas the chlorophyll molecules are also present, so that if in case the high energy light is not coming, the chlorophyll molecule will also participate in the absorbing the light from the sun. In addition, they also contains the vitamin K1 iron sulfur cluster containing molecules such as ferredoxin and the plastocyanin, which is required for relaying and running the electron transport chain downstream to the electron acceptors. And because the light energy is being used to do photophosphorylation reactions, the photosystem 1 is actually participating in the cyclic photophosphorylation reactions, whereas where it does not require the assistance from the